Welcome to 13 Universal. This is A-level biology, and we are on the last series. Okay, as you wait for your exams, you prepare towards your exam. This is immunity, series 5. I wish you the best of luck. This is Shedro from 13 University. We all are shouting out to you, wishing you best of result, excellent result. Okay, come, let's solve some questions right now. Says a successful, let me get pointer, right? Yeah, so a successful vaccination program provides a level of immunity where the majority of population is vaccinated. Okay, and it says there are several factors that affect the success of vaccination program. Which row correctly shows the factors that affect the success of a vaccination program? You can see that. That's a frequent mutation of pathogen. All right. But before then, I'd like you to try to solve the question. Okay. Yeah. Let's. Of course, you see when you see the tick there, tick means it affects. X means doesn't affect. Okay. So frequent mutation of pathogen, does it affect it? Right, but pause it and look at it. Yeah, frequent mutation affect it. Right, so yeah, these three are on the go. We cancel this. Yeah, now vaccination from 8 weeks old really is not a factor. Okay, that selects B. Why is not a factor? Because the 8 old weeks, of course, gets vaccination, but this 8 weeks old baby, okay, is still sucking mama's breast, so is enjoying what we call natural passive immunity. Okay, and of course, we're giving this baby vaccination. The immune response are still all right. Starting because the baby has to be exposed to these pathogens in order to build their own active immunity. So it's not a factor. Okay, so of course, you see pathogens are able to invade T cells. Yeah, so yeah, it's able to invade it. All right. Okay, it's a factor. That means it's going to affect the immune system. Booster vaccination needed frequently, yeah, for people that have low intake of protein. Okay, they will need booster immunity. So you see, from beginning we have chosen B. All right, so clear. Let's go to the next question. Which cell type causes secondary immune response? See, is it charge of secondary immune response to be much faster, see keywords, than the primary immune response? Which one? Okay, pause the video. Which one, okay, is what makes secondary immune response much faster? Which one? Choose it. Okay, pause the video and try it. Helper T lymphocyte, nope. Yeah, that's memory cells. Keeps memory. It keeps a track record of the particular pathogen that was in the body before. Secondary immune response means this is the second time or more that this particular pathogen enters the body. So the first time it enters the body, memory cells are formed. So memory cells are going to help for rapid response. Okay, and not this, not that. Is that okay? All right. So they keep the track record. Tetanus is a bacterial infection. Okay, let's see. A graph shows the blood antibody concentration of two people. Antibody on day 0, person G was injected with antibodies to the tetanus toxin. So they were injected with antibodies. Remember, G was given antibodies. All right, G antibody, and and person H was injected with vaccine for tetanus. Vaccine contains the pathogen itself. Okay, or the antigen. All right, let's see what we've got there, guys. Yeah, that's the graph. So what could be the result if G and H were infected with tetanus bacteria on day 20? All right, so pause the video and try it. Okay, pause the video. Remember, guys, that G was given the antibody. So G did not undergo any immune response. Okay, and it's of course passive immunity. That's passive, no immune response, no memory cells form. Okay, so passive. Is that okay? Passive immune response. Yeah. YH, all right, took some time. Okay, see, that's active. So immune response occurred because vaccine was added. So memory cells were formed. Right. So H was undergoing primary immune response here. Primary. So at this point it was at day 0. Okay. So it was undergoing primary. While person G wasn't undergoing any immune response because no antigen was injected, only the antibody from another organism. Okay. So now they said on what would happen, what could be the result if G and H were infected with tetanus bacteria on day 12, day 10. So you can see that when this was injected in day 0, okay, around day 10 or day, okay, this should be around day 12, okay. So you can see that it reaches high concentration. Yeah. All right. Yeah, that's the first time. But if they are being injected again on day 20, so if both of them are injected in day 20, so H will be undergoing secondary immune response, while G will be undergoing, right, primary immune response. Okay, let's go to the options. Tetanus antibodies would not be produced in person G. Okay, of course it will be produced. Okay, yeah, just like it was produced in person H when the tetanus injection was, okay, given to person H. So B says antibody production would peak after day 32 in person G. Yeah, remember that when H was injected at day 0, it peaked at 22 or thereabout. So there's a chance that if you inject G, that's going to peak around that point. That is just like about 12 days. Yeah, between 12 days. So from 20 to 32 is about 12 days too, because it proved in H from 0 to 12, right? So for G, if you give it to G on day 20, so it's going to peak at that. Is that okay? Because it proved that 12 days interval is enough for it to peak from the first one. Now, of course, antibody concentration would stay constant in person G. To not stay constant, it will of course peak and then drop. A second peak would occur in person G that would be lower than the first peak, right? Person H, a second peak would occur in person H. Now if you give person H the same tetanus vaccine, right, that will be acting as secondary immune response, and in secondary immune response more antibodies are produced, so it's not going to be lower than the primary. So this is also quite wrong, because secondary immune response produces larger quantities. All right, let's go to the next one. The diagram shows immune response following infection by a virus. All right, you can see that. Yeah, which role identifies the cell labeled EF? Okay, let's go. Now you can see activated. See the activated, right? Now you can see this cell here. This cell divides into two. One produces antibodies, the other one does not produce antibodies. The one that doesn't produce antibodies, pause the video and solve it. Okay, pause the video and solve it. And the one that does not produce antibody is the memory cell. So it's not plasma, it's not killer, right? It's memory. So we have these two, A and B. Let's focus on them. Then of course H should be plasma cell, right? If we check A and B, H is plasma cell. Okay, yeah, because plasma cells secrete antibodies. That's why we said that H, plasma cells secrete antibodies. All right. Now of course F produces chemical stimulation. F should be T helper. So if you look at two of them, A and B, so this shows T helper, because T helper secretes cytokines, right? T memory doesn't secrete cytokines, so that makes A the answer. Is that okay? And of course, if you check G, G is killer. Killer binds to the cell, infected host cell, and busts it. Okay? So the answer is option A. Right. Monoclonal antibodies are used to test for the presence of hormone HCG in the urine of a human female during early pregnancy. Okay, so what something is missing here. They're just asking us the one that is correct. Which statement is correct? Okay, this is which statement is correct. All right. So which statement is correct? That's what I'm trying to say. Okay. Now look at it. Number one, pause video and tick the ones that are correct. One says HCG is injected into a mouse and plasma cells in the mouse produce antibodies specific to HCG. Okay, this is what happens during the event, okay, of producing monoclonal. You inject the antigen. The antigen is HCG. You inject it into a mammal, small mammal, and then immune response will occur. Of course, plasma cells will not be produced. Okay. Now, 2 says antibodies are extracted from mouse and then fused with cells to produce hybridoma. Well, this is wrong because what is extracted is plasma cells. Plasma cells are extracted and fused with cancer cells, not antibodies. Now, 3 says single hybridoma cells are cultured and then divided by mitosis to produce a clone of hybridoma. That's called cloning. Yeah, this is correct. Okay, so it's 1 and 3 quickly. Yeah, what are functions of T lymphocyte? Okay, what are the functions? Pause the video and try it. All T lymphocyte production of cytokines. Yes, this is by T helper produces cytokines, and T helper is a type of T lymphocyte. Okay, production of toxins. This is for T killer. Is a type of T lymphocyte. Recognition of antigen bound to an antigen presenting cell. Both of them can do that. So all of them are the functions of T lymphocyte. What is correct about the role of memory cells in long-term immunity? Okay, yeah, so pause the video and try it. Long-term immunity, the role of memory cells in long-term immunity. Okay, now it says they divide to form plasma cells and memory cells when the body is attacked a second time. Yeah, that's. Now they produce a fast response so that the person infected with the pathogen does not become ill again. That's correct. Now 3 said they produce more antibodies than were produced during the primary immune response. Okay, yeah, but you can't say that they produce memory cells directly. You can say that they divide to form plasma cells before producing. It's the plasma cells that produce antibodies, not memory cells. But memory cells can differentiate to form plasma cells. Okay, because it's not direct. They don't produce antibodies directly. They have to form plasma cells, which produces. That's why we can't choose this. Now, 4 says they remain in the blood. Okay, and lymphatic system after pathogen has been destroyed. Yes, they remain there for a long time. So it is 1, 2, and 4. That is bravo, Mr. Bravo. You are the answer. All right. Which event results in a person developing actively acquired immunity? Active means, you know what it means, that they have memory cells. They can make their own antibodies. Memory cells can differentiate into plasma cells, which makes antibody. Okay. Now, the ones that can make antibodies by themselves must have been in contact with the antigen of the pathogen or pathogen of itself. It must be in contact with the antigen or pathogen itself. All right, pause the video and try it. I've given you clues. Okay. Now, becoming infected with TB bacteria. Yes, it will give you it will form memory cells, and that gives acquired immunity. Okay, it will allow immune response to occur. Drinking breast milk will give you antibodies, okay, produced by the mom, okay, and then that is passive, it's not active. Receiving an injection of antigen, yeah, that will give you, because it contains the antigen, it will stimulate immune response. Antigen stimulates immune response, and then memory cells are formed. That gives you active. Receiving an injection of antibodies. No, antibodies, right, do not stimulate, do not give acquired active immunity. Okay, it is the antigen. Antibody gives passive immunity. So it's just 1 and 3. Let's go. C. What explains why monoclonal antibodies can be used to target cancer cells? Why can they target cancer cells, monoclonal. Pause the video, right, and solve the question. Pause it, okay? A says cancer cells have different antigens from normal body. Yes, that's the answer. So they have different antigens. So when we use the antigens from cancer cells and make monoclonal, all right, remember that antibodies are highly specific because cancer cells have different antigens than that of normal body cells. So we can use it to identify or diagnose cancer cells. So A is the answer. But why is B wrong? Specific cancer drugs can be attached to the monoclonal antibodies. Remember, what we're looking at is what explains why monoclonal antibodies can be used to target cancer cells. So why can it target? It's because they can bind to antigen. Yeah, this is one of the reasons, okay, but it's not why it can target cancer cells. That's called selective toxicity. Why does it have selective toxicity? Because it can only bind to cancer cells, all right? It doesn't bind to other cells. Why? Because cancer cells have different antigens. Now, they secrete a type of antigen that binds to a specific antibody. Nope, they don't secrete. Monoclonals don't secrete the antibodies themselves, okay? This says they are secreted by hybridomas of cancer cells and B lymphocytes. This is not the reason, okay, why they can target, why they have selective toxicity. They act as snipers. Now, snipers are people that take away people in a crowd, in the midst of multitude. These snipers are trained with gun to just shoot one person. That's called selective toxicity. The same thing antibody does because it can only bind to antigens on cancer cells. All right, okay, yeah. So let's go to the next question. Yeah, let's go to the next question. All right, next question. Which statement correctly describes lymphocytes? Okay, lymphocyte. Yeah. So pause the video. Look at it. Which one describes? Say each B lymphocyte has the ability to make several types of antibodies. Okay, that's wrong. Can only make one type. Some B lymphocytes and T lymphocytes become memory cells. That's correct. Yeah, so that secondary immune response can be rapid. Plasma cells secrete antibodies. Yep. Some T lymphocytes stimulate macrophages to kill infected cells. Yes, that's T helper. Okay. So it's 2, 3, 4, guys. All right, let's go. A country introduced a measles vaccination during a measles epidemic. Okay. It was realized that vaccinated children were more likely to survive childhood than unvaccinated children, even when there were no measles epidemics. Okay. The vaccine had given the children some protection against other pathogenic infections. Which statement, the vaccine for measles had given children protection against other pathogenic infections. Which statements could account for this extra protection, right? All right, pause the video and look at it. A says B lymphocytes produced memory cells which gave the children passive immunity to these infections. B lymphocytes produced memory cells which gave the children passive. It didn't give them passive. Memory cells give active. Okay, so active, right? If they come in contact with the antigens, to be active immunity, not passive. B says memory cells produce plasma cells which secrete anti-measles antibody that bound to antigens that closely resemble measles cells. Okay, this makes sense. All right. B says memory cells produce plasma cells which secrete anti-measles antibodies that bound to any antigen. No, antibodies are known for specificity. So this is wrong. T lymphocyte produced memory cells which gave the children natural immunity against these other infections. No, that's not natural. That's artificial. Okay? It was not due to natural infection, cause of infection. It was due to vaccination. Right, so B is the answer. A patient with kidney failure can be given a kidney transplant in a kidney transplant healthy kidney is taken from another person and put into the patient's body. Sometimes donated kidney cells are killed by patient's body. In this primary response, immune response, T lymphocytes bind to antigen on donated kidney cells and they are activated. The statements describe the next stages of the primary immune response. These are the next stages, okay? Let's see what goes on there. Which order correctly explains how donor kidneys are killed? How are they killed? Okay? A says 1. Activated T lymphocytes secrete cytokines. Yes. Yes, so A and B. So that's where to start, because B and C said T killer cells bind. No, they don't start there. Start from, alright, T helper cells, okay? Secreting. Because T killer cells bind to antigen on donor. No. Alright. Okay, let's check the ones. Okay. Of course, after that, then T lymphocyte multiply by mitosis and differentiate into T killer. Yes. Right. Then what happens? T killer cells then bind to, right? T killer cells bind to. That's the next one there. Look at it here. Yeah, that's 2. Now, 2 says B lymphocyte differentiate into plasma cells that make antibodies. Well, 3 says T killer cells bind to antigen on donor kidney cell. I think it should be 3. Why? Because we're looking at T lymphocyte, okay, not B lymphocyte. So then 4 says T killer releases toxin that destroys donor. So this is on T lymphocyte response, not on B lymphocyte, okay? That is why we cannot choose 2. 2 is on B lymphocyte, okay? We're looking at T lymphocyte. Alright, the diagram shows the simplified structure of an antibody. Oh, that's an antibody, guys. Which statement is correct? Okay, which statement is correct? Pause the video and try it. U and X allow the antibody to bind to different antigens. U and X, they are the same, okay? They are the antigen binding site, the variable region, so they bind to the same antigens. So they don't bind to different antigens. You can check, they have the same tertiary structure, okay? So A is wrong. V says allows the antibody to be flexible. V allows the antibody, this is the hinge region. Yeah, it allows it to be flexible to bind to antigen. This is correct. W can bind to one specific antigen. Nope. That's the constant region binds to, right? It can bind to one specific antigen. X can bind with specific antigen, specific phagocyte receptor. Nope. This is the one that can bind to phagocyte. So the answer is bravo. Alright, which statement about uses of monoclonal antibodies are correct? So pause the video and trash this question. Monoclonal antibodies can be injected into patients to give active immunity. No, antibodies don't give active immunity. When you inject into them, it is when you inject antigens that trigger immune response, leading to formation of memory cells that produce active. It can only give passive. Now, 2 says monoclonal antibodies can be injected into patients to treat viral infections. Yes, that could help. Monoclonal antibodies can be used as diagnostic tool for antigens. Yes, because they can bind to specific antigen on the pathogen. So, C is correct. Which statement explains how a vaccination program can control the spread of an infectious disease in a population? Alright, pause the video and try it. So unvaccinated individuals are less likely to meet vaccinated, okay, individual. That's not how it is controlled, right? It's not about vaccination. Okay, this is what okay, look at B. Unvaccinated individuals are less likely to meet yes. So if you vaccinate almost everybody, so unvaccinated people are less likely. That's what we call herd immunity. Herd immunity, right? Alright. Then C says vaccinated individuals cannot catch the disease. They can catch it if they were not given booster. Vaccinated individuals have natural active. No, they have artificial active. Okay, so the answer is bravo. Yeah. Let's go to the next one. Which blood cell types do not they do not recognize, they do not engulf, and they do not digest non-self, okay, particles. They do not digest. Right? Pause the video and try it. Macrophages digest, neutrophils digest. They are all phagocytes to digest, but T killer does not digest. Okay, so this is not the answer. That's not the answer. We're looking for the one that does not. The one that does not. Okay, this is D. Delta. Repeated infections with malaria. Repeated infections with malaria. Okay, result in more effective immunity to malaria. Which types of immunity is responsible for the more effective immunity? All right, so pause the video and try it. So which types of immunity is responsible for more effective immunity? Okay, malaria, infection of malaria, this is natural. Okay, but because it involves the pathogen, so it's going to trigger immune response. Pathogens always trigger immune response. They're antigen, and memory cells are formed, and then active immunity is formed. So it's going to be artificial active. No, not artificial. It's natural active, because it's due to natural cause of infection. So it's not going to be artificial because it's not man-made. Okay, repeated. It's not passive because no antibodies from another organism are injected. It's not natural passive because no antibodies, but it is natural active because there is direct exposure with the pathogen and the antigens, which causes immune response to form memory cells. Okay, T lymphocytes have a protein PD-1 on their surface. Some cancer cells have a receptor molecule on their surface which binds with PD-1. So you see, T lymphocytes have PD-1. Cancer cells have they have what? Receptor that bind to PD-1, inactivating the T lymphocyte. So these cancer cells bind to PD-1, they inactivate the T lymphocyte. A monoclonal antibody lambrolizumab has been produced against this receptor. Okay, trials show that 54 of 135 people with advanced skin cancer who were given lambrolizumab, okay, the tumors more than halved in volume. Okay, in 6 of the 57 people who were given the highest dose, the tumors disappeared. Amazing. What may be correctly concluded from this information? It's amazing. Okay, yeah, pause the video and let us know what you think. Okay, solve the question. You see, lambrolizumab binds with a receptor on the surface of skin cancer. Yes, because they told us here the monoclonal has been produced against this receptor. So this is monoclonal, so it binds the receptor. Okay, this is correct. 2 says cancer cells to which lambrolizumab is bound, okay, cannot inactivate T lymphocyte. That's correct. It can't inactivate it. Okay. Now 3 says lambrolizumab targets and kills cancer cells. That's not it. Doesn't kill them. Just binds to them. Okay. 4 says lambrolizumab allows a patient's own immune system to kill cancer. Yes, it just prevents cancer from in activating T lymphocytes, and then the patient's immune system can now kill it. So it should be 1, 2, and 4. All right. Now, a person's blood group is determined by antigens present on the red blood cells. Can see that. Yeah. Okay. So this is it. So what is the question? The question says, during a blood transfusion, it is essential that a person receiving the blood does not have antibodies to the donors. Okay. And which blood groups can be given to a person with blood group AB? So AB, right, has the antigen A and B. Okay, so they have no antibody against anything because they already have the antigen, right? So these antigens are self, they don't make antibodies against itself. So that means they will receive from everyone. Okay, they are called universal recipients. Okay, are you an AB, right? That's amazing. Which types of cells are stimulated to divide by cytokinesis? Okay, by cytokines produced by T helper. So the ones that divide by cytokines. So pause the video and try. Macrophages, yeah, but it's not the only one. B lymphocyte only, no. T killer cell, no, but see, B lymphocyte and T killer, they are together here. So we go with D. Okay, they are stimulated to divide. B lymphocyte make divide and then make antibodies. Okay, while T killer, all right, burst infected body cells. All right. Alright, see that a blood cell count can indicate how many white blood cells there were in the blood. Whereas in the body are white blood cells found. Where are they found? Okay, where else are they found? Okay, pause the video. Why do you find white blood cells? Some of them, of course, are found. They are found everywhere. Okay, yeah, found everywhere. A monoclonal antibody specific for a virus was treated with an enzyme to break the bonds between the variable regions. So if this is an antibody, guys, see, yeah, this is an antibody, Y shape. This is the variable region. We have seen short chain joined to long chain. Okay, that forms variable region, short chain joined to long chain. Okay, and this is this. So they treated with enzyme, all right, to break the bonds between the variable region and constant region. Okay, so they break the bond here, the bond between them. This is constant region. Okay, every here. So these are the variable region, this and this antigen binding site. If we break the bond between them, it's carried up like this. Okay, and then they says they separate these separated variable and constant regions. So if we break it, we separate them. Okay, well then added to cells infected with virus. Which statements are correct? So pause the video. The first one says the constant regions would bind to different parts of the viruses. Constant regions bind to, all right, to bind to different parts of the virus antigen. Constant region. So what do you think? All right, I think you should bind to, all right, one part. All right, let's see. The viruses could be engulfed by phagocytes if they were present. They could not be engulfed because we are broken, right? Yeah, we are broken the antibody. Antibody helps to tag or mark out the viruses. Is that okay? All right. It says, 3 says the variable regions would all bind to the same part of the virus. The variable binds to the same part. Okay? So the constant regions would bind to different parts of the virus antigen. So what do you think is the answer? Of course, this is the only correct answer there. All right, let's go to the next question, guys. Which role correctly describes the type of immunity gained from being injected with antitoxin? Yeah, antitoxin. Remember, you are the one injecting it, so it is artificial. Which one is artificial? A and C. Okay, because it's an antibody, so it cannot be active. So active and natural out. So it's artificial passive. All right, that's C. All right. Yep. Let's run, guys. Where are antibodies found during an immune response? So pause the video. Where are they found, guys? Remember, during an immune response, you know, we see having clonal selection, clonal expansion, and then differentiation, and then of course different responses. So by that time, antibodies may not have been formed. So they are neither on the surface of pathogen nor in the blood plasma. They have not been formed because antibodies still going on. This during an immune response. Okay, so it is when the immune response is done, then antibodies can be made, right? All right. So during immune response, antibodies are not even made. So Of course, they could bind to the pathogen. They could also be secreted into blood plasma. They have not been made yet, so that's why we choose D. All right. So, say the statements are about infectious diseases. So I want to solve this. Solve this. So, what is the question? Which row matches the correct statement number with each disease? Okay. Now, this is cholera. Which one fits cholera? Say disease may be spread between humans by contact. No, cholera is not spread by contact. So A cannot be the answer. B cannot be the answer. Okay, let's check 2. Disease causing organism has many antigenic variations. Cholera doesn't have many antigenic variations. So C cannot be the answer. So D is the answer. Though we have to look at this sentence. A vaccination program is available. Yes, there's a vaccination program for cholera. Now we have chosen this. Let's test for measles. Now, this option says that 1 and 4 are good for measles. 1 says disease may be spread between human contact. Yeah, measles spread by human contact. 4 says, yeah, there's a vaccination program for measles, so that's fine. Malaria, 2 and 3, disease causing organism has many antigenic variations. Yeah, the disease causing organism is a eukaryote. Plasmodium is a eukaryote. It has genetic complexity, it has many genes coding for many proteins that act as antigens. So 2 is right for malaria. 3 says spread of disease may be influenced by climate. Yeah, it's endemic in the sub-Saharan Africa because of the temperature and humidity. So it's found in a particular place. The rainfall in sub-Saharan Africa is high, so stagnant waters are formed. The temperature is high, so that helps the pathogen to develop to its next stage of life cycle. So that 2 and 3 works for malaria. Okay, so the answer is the So thank you guys. Wish you the best of luck in your exam. This is where we're stopping. Watch all our videos, alright, and make sure you practice this question. When you get into exam hall, start with the questions you know first, the topic you know first, okay? Leave long questions and do them last, right? So this is Shedro from That Universal. Wish you the best. Bye.